Hello students, welcome back to my channel. In this video, I am going to explain you about some interesting fact that takes place in our cell, that is translation. In general, translation means something written in a text. It should be converted in our own language so that we can understand its true meaning. Likewise, in a cell, the body is able to make a protein by getting the information from DNA. So, in this video, I will explain about the translation process that takes place in our human cell. Stay connected with me to understand more about the facts. In a cell, we know that inside the nucleus we have DNA, and DNA is the functional unit of a living cell. It has all the informations. So the information from the DNA is copied into mRNA, and this process we call it as transcription. So in the last video, I discussed detail about what do you mean by transcription and how the DNA is copied into mRNA. So in this video, we will learn about that the copied mRNA, the information in the mRNA is translated into a functional protein, and how this translation takes place, how it is converted into a functional protein, we will learn detail in this video. So what are the things first required for a translation to take place in a cell? The first one is messenger RNA. Messenger RNA is nothing but it carries a genetic code from DNA in triplet that we call as codons, which specify particular amino acids, because amino acids are the building blocks of proteins. Next one, ribosomes. We need ribosomes. That is a cellular machinery. It has both small and large subunits, and this is the location where the translation takes place. Next one is tRNA, that is transfer RNA. These are the molecules with anticodon on one end and a corresponding amino acid on the other. It acts as an adapters. And last, amino acids. We need amino acids because amino acids are the building blocks of the proteins. This picture clearly shows the components required for translation. The first one is mRNA. You can see the codon here. So, triplet codon, the three codons in the mRNA code for a particular amino acids. And this is transfer RNA, which has anticodon in one end and the other end carrying the amino acid. And this is the ribosomes having large and smaller subunits. So we can learn in detail what are the importance of these mRNA, tRNA and ribosomal RNA in translation. First we learn about ribosomes. What do you mean by ribosomes? So we have two types of cells, prokaryotic cells and eukaryotic cells. So the ribosomes is also of two types. In prokaryotic cell you can see 70S type of ribosomes, and in eukaryotic cell you can see 80S type of ribosomes. And the larger subunit it is 50S and the smaller subunit it is 30S in prokaryotic cell. And in eukaryotic cell it is, for the larger subunit it is 60S and for smaller subunit it is 40S. And also it has the RNA, single stranded RNA in each subunit, and the RNA strand will be different. Here it is 23S or RNA in 50S larger subunit of prokaryotic cell, and here also you can see in 60S larger subunit of eukaryotic cell you can see 5S RNA, 28S RNA and 40 different types of proteins. So the ribosomes is made up of RNA and both proteins. This picture clearly shows the location of the ribosomes. So clear picture of the ribosomes. So when the translation has to be takes place, the mRNA binds with the ribosomes, the small unit, and the larger subunit will have the A, P and E site. And what's the function of this A site? A site, it receives the transfer RNA. P site also will have the transfer RNA. When the bond is formed between two amino acids, it exits from the E site. So in upcoming slides we can learn in detail about the importance of this sites in the ribosomes. Next one is tRNA. So tRNA, the structure of tRNA you can see like this, a loop like structure, and it has D arm, a variable arm and TY arm and an acceptor arm in the top. In the down it has anticodon arm. See, this is the anticodon regions. And in the top we have the another region, it is called as acceptor arm. That means it accepts the amino acids. Anticodon means nothing but mRNA. We say the triplets, we call it as codon, and alternate to this we call as anticodon. So it gets the information from the mRNA and accordingly it brings the amino acid to the region. So this is the function of the tRNA. It acts as an adapter between the mRNA and the amino acids. So what are these amino acids? There are 20 different amino acids in our body. So this tabular column, it shows the 20 different amino acids and the 3-letter code. It can be represented as alanine, means it is represented as ALA. In a single letter code, it is represented as A. So the amino acids are alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, triosine, valine. So these are the 20 different amino acids which we can see in our body, and the methionine is always the start codon. That means when the translation has to be started, the first amino acid will be the methionine. Then comes the other amino acid according to the codons present in the mRNA. See, to understand, you can see here. So the triplet code, already I told that the mRNA has a triplet code which gives the message for the amino acids which has to be brought to the ribosomes to form the proteins. So if the codon is UUU and UUC, it codes for PHE, means what? Phenylalanine. Okay. And now see here, UUA and UUG. If the codon has this type of the arrangements, it codes for leucine. LU means leucine. Okay, and I already told AUG, it will be always the start codon in the mRNA. It always represents the methionine. And we have the stop codon also. If the codon has UAA, it is a stop codon. That means after that there is no translation. That means there is no addition of amino acids. And UAG is also the stop codon, and UGA is also act as a stop codons. You can see here the amino acids, the different types of amino acids, they bind together to form the proteins. So the bond between the two amino acids we call as the peptide bond. Now this is the DNA which has the codons. So the information from the DNA is first copied into mRNA. You can see here. So the 3 codes, that means GAG, it codes for glutamine, the protein. CCC, it codes for proline. So the 3 codons combine together to form an amino acids. It codes for the amino acids, and the amino acids, they form a peptide bond to form a protein. So only we call it as protein as a polypeptide chain. Hope you understood this. Yes. Now the translation, it takes place in 3 steps, that is initiation, elongation and termination. So for the initiation, the codons should recognize the translation, it takes place in 3 steps. The translation, it takes place in 3 steps. The first step is initiation, second step elongation, and third step is termination. So the translation to be initiated when the mRNA binds with the ribosomal RNA, the transfer RNA has to recognize the start codon. So only the attachment of amino acids takes place with the help of the tRNA. To recognize the start codon in eukaryote, in prokaryote, we have 2 different types of translation. It takes place in 3 steps: initiation, elongation and termination. So the translation has to be initiated. The tRNA should recognize the start codon. So once when the mRNA gets attached with the ribosomes, the tRNA has to recognize the start codon. To recognize the start codon, in prokaryote we have some sequences, see, that we call a Shine-Dalgarno sequence. So that will be the repetition of UAAG, GA, GGC, GGU, you can see here. So this sequence we call it as Shine-Dalgarno sequence. So after this, it can easily recognize the start codon. So once it recognizes the start codon, AUG, it always codes for methionine, and then the tRNA brings the amino acids related to these codons. As well as in prokaryotic, we say it as Cossack sequence. So once when the mRNA is attached with the ribosomes, there is some sequence you can see here, GCC, GCC. So these are the sequences. When the tRNA sees this sequence, it can recognize that. After that, you can see the start codon. See AUG, you can see here. So in eukaryotic, the sequence we call it as Cossack sequence, and in prokaryote we call it as an Shine-Dalgarno sequence for the initiation of the translation. This picture clearly shows the elongation of the translation. So already we discussed about the structure of the ribosomes. So it has A, P and E site. So the first transfer RNA having the amino acid methionine gets attached with the start codon AUG. So it is located in the P site. So the next amino acid comes here according to the codon present in the mRNA. The transfer RNA brings it and gets attached with the A site. So once when attached to the A site, the bond, peptide bond will be formed between these two amino acids, and the mRNA it moves. Ribosomes also it moves. So when it moves, this two transfer RNA it comes to the E site. First one, this amino acid comes here, and next this comes here, and it get exited from the ribosomes, and next transfer RNA it brings the amino acid related to the codons present in the mRNA, and we can see a bond which will be formed between the amino acids, and at last we can see a complete polypeptide chain. So once the signal it gets, that means C stop codon, when it reaches the sees the stop codon in the mRNA, the transfer RNA it stops bringing the amino acids, and we can see a release factor, that is something which blocks the translation. And once the translation is completed, it gets the amino acids get detached from the tRNA and the ribosomes, and it enters into one functional one. This picture also clearly shows the formation of proteins. Hope you understood this. So as a summary, you can see here. So in the mRNA, we will have the codons, and the tRNA will have the anticodons. According to this, the amino acids get attached to the mRNA. So in the ribosomes, once the mRNA get attached with the small subunit of the ribosomes, in the larger subunit, you can see the transfer RNA. The first codon is always AUG, which codes for methionine. And once it gets attached, the tRNA, it brings the amino acids related to the codons present here. See UAG. So here you can see that AUC, anticodons and the corresponding amino acid, and it comes in the A site. So once a bond forms between the first two amino acids, the transfer RNA get detached from the ribosomes from the E site, okay, leaving the space, and then the mRNA it grows, so that the transfer RNA it reads the other codons present in the mRNA, and we can at last we can see a complete formation of a polypeptide chain that we call as the proteins. And how this polypeptide chain is formed. So when you see the structure of the amino acid, it has carboxyl group and an amino group. So in between the carboxyl group and the amino group, the water molecule is removed. Once the water molecule is removed, a bond will be formed between nitrogen and carbon in the group. Okay, so this only we call it as a peptide bond with the removal of water. So one by one the amino acids get attached with each other to form a protein. Now, why the proteins are important? In the last video also, I explained about the importance of proteins. The proteins are very, very important in our cell, because our cell, our whole body is made up of what? Proteins. The proteins are essential for the transport of blood, hemoglobin in our blood, central nervous system, wound healing, regeneration, for our fluid balance, acid-base regulation, hormones, enzymes, immunity, structure and mobility. Everything this made up of what? Proteins only. So the which protein has to be formed, and when a protein is required, the information we get from the DNA, and the DNA it is copied into mRNA, and from the mRNA the protein will be translated. Okay. And there are different structures of proteins also. So hemoglobin, the heme group which we see in our blood, it is the quaternary structure of the protein. And the antibody, that is in immunity, the antibodies, the structure of the protein will be the tertiary structure. And this is the collagen for the structure. Here it is the alpha helix. A quick recap of what we learned today, that is about translation. So Inside the nucleus, we have the DNA. The information present in the DNA is copied into mRNA inside the nucleus, and once it is copied, the mRNA, it is transferred to the cytoplasm, and in the cytoplasm it gets attached with the ribosomes, which has two subunits, smaller subunit and larger subunit. And once it is attached, the translation takes place, that is attachment of the amino acids with the help of transfer RNA. So according to the codons present in the mRNA, the transfer RNA brings the amino acids with the anticodons, and between two amino acids a peptide bond will be formed, and once it reaches the stop codon, translation will be stopped, and the amino acids which are bind together, they form a protein. So only we call it as the proteins or the polypeptide chain. So whenever our cell requires a particular protein, the information from the DNA is copied and the process is taken place, and at last the body gets the proteins. Hope you all understood the translation. Thank you. Thank you for your listening and happy learning. Stay connected with Bio Wonders to learn about many interesting facts about biology. Thank you.